know that dairy cows require a lot of energy to maintain body condition and maximize milk yields. One way to help meet those needs is to offer high-quality silage, which provides an abundant source of energy, protein, and fiber that aids rumen health. The process of making silage involves allowing grasses and legumes such as alfalfa to undergo fermentation in an air-free environment. Various foliage crops can be used to make silage, and when successful fermentation occurs, it results in high quality and nutritious feed that can be safely stored for many months. Why make silage and not just offer hay? Fermentation helps to preserve more nutrients. Now, we need to stress the importance of quality silage here. Sometimes things can go wrong during or even after the fermentation process, such as the silage being exposed to air. When this happens, the silage can become contaminated by fungi. Fungi can spoil silage, making it less palatable and nutritious. In some cases, fungi can also produce mycotoxins, making the silage dangerous for your cows to eat. What are mycotoxins exactly? Mycotoxins are chemical compounds produced by some species of fungi. When consumed, they can cause issues including gastrointestinal upset, decreased appetite, reduced weight gain, and even organ damage, making it something we want to avoid in our silage as much as possible. However, because most farmers produce and use their own silage on farm, not a lot of data exists to tell us about the levels of silage spoilage across Canadian dairy farms, or even what kind of fungi are living in silage on farms across Canada. Recently, a study was done attempting to fill this gap and learn more about fungal spoilage and mycotoxins in silage on Canadian dairy farms. The researchers worked with over 100 farms across Canada, looking at samples of first-cut grass and alfalfa silage. The silage they looked at came from different kinds of storage systems, including wrapped round bales, tower silos, and bunker silos. DNA sequencing was used to identify the species of fungi present in silage samples, and chemical testing was used to determine which mycotoxins were present and in what quantities. The results showed that the most common fungal strain was from the genus Monascus, which has also been found in corn silage. Research also shows the presence of Monascus in silage samples from other countries around the world. Other types of fungi included penicillium and yeasts such as Pichia. Types and abundance of fungi generally did not differ between location and type of silage storage, except for in Atlantic Canada, where a small number of fungi were more commonly found in towers than in bunkers or in bales. The researchers found that 94% of the samples taken included species of fungi that have the potential to produce mycotoxins. However, fewer than 10% of samples actually contained detectable quantities of the mycotoxins that were screened for. These results suggest that while fungi with the ability to produce mycotoxins are present in feed samples, they generally were not actively producing those mycotoxins, and therefore the risk to cattle health is fairly low. This study shows that high-quality silage can be made using different types of silage systems, with limited differences in the risk of fungal and mycotoxin contamination and low overall risk to cattle health. Research on fungal spoilage and its impacts on silage quality is important for dairy farmers to know. Many Canadian dairy farmers make and store their own silage, so better understanding the types of contaminants that may or may not be present is important and allows farmers to make the best feed possible for their animals. While dairy farms and silage systems vary across Canada, these findings contribute to research from different regions and silage types and will help to inform future management recommendations for Canadian dairy farmers.